Okay, right. Happy start කරමු නේද? Right. So good afternoon. So I think few of you might have been must be still on holiday. So it doesn't matter. I mean, because I had to give a holiday last week, I had to do the class today. Right? So I hope none of you came and went back, right? Nan, you all of you got the message. So the key point is the group, okay? Don't mute the group or do anything, okay, right? The group is there only for urgent notifications. So all of you are in a group, right? Headway group is there, and for the online people there's a community and other groups are there, right? So I won't be spamming the groups, and also the permissions are limited, so it's mostly me and the hotline, that is hotline number that is sending messages. So always pay attention to what I send to the group. Okay, nikan diagram එකක් හරි, just a small text message or a video or whatever I send to the group is very important. Okay, so urgent notifications and messages are sent to the group. So for all of you and also for online people, everyone, always be alert of the groups. Okay, so all the things will be updated, and the tunes will be sent, and everything will be happening through the groups. So pay attention to those, right? And so we can continue from where we stopped last week. So the plans, upcoming plans in the class, right? So I think I sent you the April month plan, right? So for you and for my Nugegoda class, almost the same plan but different dates, right? What's going to happen is today we are going to finish proteins and also start nucleic acids, okay? And then next week it's going to be the practical session, okay? It's going to be a practical session from carbohydrates, proteins, lipids, and I'll try to, it will cover a microscope section as well, okay? So microscopes is going to be the first part of the new lesson, okay? That is not new lesson, the new segment of this lesson. මම කියවනේ මම මේ lesson එක කැලී ටිකකට කඩනවා කියලා, right? So the first part is biological molecules, water, carbohydrates, මේ කරගෙන යන set එක. Then it's going to be cellular structures. That's what we are going to start. Okay, cellular structures වලට පස්සේ we have cell division. Then we start a big section that is called metabolism. Okay, තියෙනවා enzymes, photosynthesis, cellular respiration. Okay, there are few topics are there. So for all that, there's going to be a new tute. Okay, there's going to be a new tute. This I will distribute next week. Okay, right? So this is our old tute. Make a wage neve, right? So these tutes are going to follow the structure of my typical tutes. Again, normally then a tute pidira thamai dayanne. That tute was a bit special because you didn't have the resource book, right? So I had to put all the resource book content in the tute as well. So this point onwards, I won't be giving resource book content. Okay, so the first message is you have to figure out the resource book. Okay, either buy it directly from the Ministry of Education, එහෙම නැත්නම් get a printout. Okay, so grade 12 book එක විතරක් දැනට ඇති, grade 13 එක buy it later, take a printout later. So get the grade 13 resource book as soon as you can. Okay, that is for both physical and online people. So this new tute is going to have only diagrams. මේකේ තියෙන්නේ diagrams විතරයි, right? It's only going to have diagrams and some space for you to make short notes, right? මම දෙනවා short notes with the extra sort of bits and pieces of knowledge that you need to know, right? So it's going to only have diagrams. Okay, the මේ වගේම there's going to be another tute which is going to only have classified past paper questions, right? It has also 30 pages. It's going to have classified past paper questions. මේ වගේම තව booklet එකක් හම්බ වෙනවා that is going to have past paper questions, structured essay and essays, as well as the practical guide is going to be there as well. Practical guide. Okay, we are going to do 7 practicals, right? 7 practicals, I mean 3 major practicals, and under that you are going to have small sub practicals. එතකොට ඔක්කොම 7ක් විතර එනවා. හරිද? So that is the normal structure that I usually give for my 2027 batch, 6 batch ඔය ඔක්කොටම කරගෙන යනවා. So ඔයගොල්ලන්ටත් දැන් ටිකෙන් ටික I'm going to bring you to the normal sort of that study pattern. Harida? So we are going to be done with this first tute very soon. So very soon we are going to be done with this. So any questions? Shall we move on? Online people, can you hear me? All good? How's the new year? Right? A lot of fatty and carbohydrate food, the lipids, lipid containing food, less protein. The Sri Lankan diet very low protein containing. Have you thought about it? Right? Most of you might not be celebrating New Year, but the Sri Lankan typical Aurudu food, it's all sugar and oil, right? Everything, it's either rice flour or whatever. Sugar, you put sugar and you coat it in another coating of flour and then you deep fry it in oil. Most of it. Kyaung cookies ask me everything, right? Deep fried carbohydrates. So oil and all the sort of the negativity. So if you take, priority of nutrients, you can just survive on proteins. The essential nutrient is protein. Protein, if you take a protein containing food such as eggs, eggs contain mainly protein, then it has cholesterols, lipids and also a bit of carbohydrates, right? So other side, we take the main food is rice, which is a carbohydrate, then we add to it some oil and lipids. Protein, right? So that's why we have a risk of non-communicable diseases, right? Higher prevalence. Anyway, proteins. Where did we stop last? Proteins? Yeah. Page number 16. Page number 16. So for online people who haven't got the tute, I'll send the PDF, right? The PDF is now available in the chat, okay? Any quality issues? All good, right? So I'll remind you, right, as we were talking last week, compared to your carbohydrates or lipids, your proteins are much more complicated. That's why we are going to spend a bit more time studying about proteins as well. Because if you take your body, your proteins are the workforce, right? Your proteins are working molecules. Why do we say that? Because if you take your cell and you are made up of cells, right? A cell is like a factory, it's a working sort of a unit. In there, your carbohydrates can be either structural or storage. Carbohydrates can build up things or you can store carbohydrates. Lipids can also be energy source or structural. Mainly. But if you take functions of proteins, almost everything is done by proteins, right? For example, they can act as defensive. They can protect your body against sort of infections, bacteria. Then they can act as enzymes. They can do reactions. Metabolism is protein based, enzyme based. Okay. Then imagine you are contracting a muscle, you are lifting some weight. Proteins are working. Okay. Then again storage. Your proteins can store things such as in your eggs you have certain type of protein. Milk has protein. If you take your blood transports oxygen and carbon dioxide by hemoglobin, which is a protein, right? And also proteins can act as structural. In your skin you have proteins, right? That's why you have such a good skin protection. Right? Then your hair, your nails, in your sort of your muscles are connected to your skeleton by what? Tendons. Yeah, tendons are than chicken elastic, right? Muscles are edge tendons. Then ligaments are chicken leg galavan bones galavan, that's because they are connected by ligaments. All those are proteins. Proteins are very strong. Okay. So that's why proteins have a wide variety of functions. Okay. That's why lasium protein if you are going to make a polysaccharide, monosaccharide dehydration you get a polysaccharide. Yeah, you connect a glycerol and 3 fatty acids and you get a fat, right? Proteins are not like that. Imagine a functioning protein, like at the end you have a functioning protein like a nice birthday cake. Adana candles gahala full decorate kara nama gahala thiyena full product එක. Okay, so if you want to, I mean, to get there you have to undergo a lot of steps ne. You need to take the basic ingredients oka bake kalla ඊළඟට tattu hadala icing kalla, right? There's a lot of things to be done. Exactly, it's the exact like that. If you want a working functional protein, you need to do a lot of things. Okay, so today that's what we are going to study about by levels of protein structure. Ita kalling I'll do a small revision. What do you need to know about a protein? What the proteins gonna denagandu, right? So proteins are built up of what? What is the building block of protein? Yeah, what is the building block of protein? Yeah, proteins are composed of 20 amino acids, right? So in this earth, I mean on this earth, is there only 20 amino acids? Vissamatti and amino acids? No, there's more than 20, but out of that only 20 is contributing to form proteins. Got it? So 20 amino acids contribute to form proteins. If you take the structure of a amino acid, you need to be able to draw it, right? What are the groups? You have a carboxyl group. You have a carboxyl group, central carbon. Then there's a again. Yeah, who is this NH2 group? Amino group. Amino. There's a hydrogen. There's a hydrogen. And who is the remaining one? Alkyl group or the R group. Variable. So he is the one, this R group is the person that is determining, I mean, that is going to change these 20 amino acids, right? All these groups, amino, carboxyl, hydrogen with the central carbon, they are common. They are common to all 20 amino acids. So this is called what? Yeah, this set of groups, backbone. It's called the backbone. Okay, then what do you call this one? This lonely R group. He's like sort of sticking out. The may set home, if I connect another amino acid here and another one here, it's going to form a nice sort of a chain, right? So in that chain, who's going to be sticking out? The special one. This. Therefore, is the side chain. He's the side chain. Okay, he's a side chain. Got it? Right. So this is the basic structure of a amino acid. So how do amino acids connect to form a what? When amino acids connect, what do you get? A polypeptide. Polypeptide. Amino acids connect to form a polypeptide. Right? So how does that occur? What is the bonding? So another, imagine there's another amino group, right? There's another amino group. There's a carbon, there's a hydrogen, there's a C double bond OH, okay, and there's another R group here. Okay, there's another R group here. We have to give R2 kilo, within a different kilo. Okay, so how is the bonding going to happen? How is the bonding going to happen? It's also a dehydration reaction. Dehydration. May OH ekai, may hydrogen ekai set karana. Yeah, it's going to release a water. Then what new molecule are you going to get? What is the new molecule you are going to get? There's going to be a new bond forming here, right? There's going to be a new bond. I'll erase this, okay? Right? There's going to be a new bond forming between this carbon and this nitrogen, That is called what? What is this new bond? Yeah? Peptide bond. Peptide. Peptide bond which is formed due to a condensation or a dehydration reaction. Okay? Which is formed due to a condensation or a dehydration reaction. So this is how you get a polypeptide. This is how you get a polypeptide. Got it? I make either. Did we draw this? Page number 16. We drew the basic structure of an amino acid. Yeah? We drew an amino acid. What is the simplest amino acid? In a simple mechanical. Yeah? Who is the simplest amino acid? Yeah? Online people? Who is the simplest one? The amino acid with a hydrogen in the R group. R position just another hydrogen atom. Yeah? That is called, this amino acid is called what? Glycine. Glycine is the simplest amino acid. So, then glycine as an amino acid. The only amino acid you need to know is glycine. Right? Is that isoleucine, leucine, fumarate, no need to know them right now. Glycine is the simplest amino acid. It has a hydrogen in the R group place. That's all you need to know. Clear? Right. So, what did we talk about next? Amino acids may have one or more carboxyl groups or amino groups. So, in the place of this side chain, you can have another carboxyl group or another amino group. Amino. Okay. Then, amino group has alkaline nature and carboxyl group has acidic nature. Right. So, this carboxyl group is acidic. Carboxyl acid group. I told you, organic chemistry, right? The organic chemistry in another year's time, right? Until that, I have to teach you these things as well. Amino group has basic nature. Okay? Amino group is basic. That is because of a theory of acidity, basicity. I mean, in O levels, you think acid is something that can give a hydrogen. A base is something that gives OH. That is not, that is one way of describing acids or bases. There is only one way. There are so many other theories. There are Lewis acids, Lewis bases, theory. In that case, how we define an acid is someone who has a lone pair of electrons, that is a base. Okay, the main nitrogen, this amino group, this nitrogen has two lone electrons on the nitrogen. So we call him as a base, like that. Got it? So in organic chemistry and also when you are doing equilibrium, you will learn about these different theories of acids. Just for now, remember this carboxyl group is what? Acidic. This amino group is what? Basic. Okay. So acidity and basicity are found in the same molecule. Therefore, it's a amphoteric molecule. Okay, amphoteric. Ek api highlight karane. I have bold color dila dina. It's an amphoteric molecule. Right. So this is all clear, ne? Right? This is all clear. Two amino acids undergo condensation reaction by removing a water molecule from both and result in a bond known as a peptide bond. Make that a peptide bond. It's a what sort of a reaction? Condensation reaction or a dehydration reaction. Protein is composed of one or more polypeptide chains which are composed of amino acids. Okay. That sentence is nice. Okay. Protein is composed of one or more polypeptide chains. Okay. So you get a chain like this, no? It's a chain of amino acids connected by peptide bonds. Right? So when you are making a protein, you can have either one chain, one polypeptide chain, or you can have more. Okay? We will talk about such proteins. They are a bit complicated proteins. Okay? More than one amino acid polypeptide chains get in. So we drew this, formation of peptide bond. right? Uh, okay, so Windows is starting, right? TNT matak wenawa neda? Okay, so today we have to move on. So we now have like a basic cake, right? අපි දැන් හදලා තියෙන්නේ මොකද්ද? දැන් මෙතන හදලා තියෙන්නේ මොකද්ද? Dipeptide දැන් නේ දෙකයි. Amino acids දෙකක්. Dipeptide. So we are going to continue this polymerization process. This OH එකට තව nitrogen group එකක් connect කරලා we are going to put another amino acid here, another amino acid here. It's going to have a long chain. Okay? So that process is going to continue, and that sort of a chain of amino acids, we give it a special name. කියවන්න බලන්න. Levels of protein structure. හරිද? Levels of protein structure page 16 යටම තියෙන. Okay? There are 4 levels of structure which play important roles in their functions. හරිද? Primary structure. Primary structure. Let's read. That's a definition. හරිද? That's a definition. ඔයගලන්ගෙන් අහනවා what is the primary structure කියලා. Right? Unique sequence of linearly arranged amino acids linked by peptide bonds is the primary structure. Right? So, you can see there are how many keywords? 1, 2, 3. I mean, if you look more carefully, there's 4 things you cannot miss. What is the primary structure? When you write the definition, there are 4 words that you cannot miss. Right? It's a unique sequence. It's a unique sequence. Amino acid chains don't have a thing. Right? Amino acid chains, polypeptide chains are different from one another. Right? Then collagen hadana amino acid chain එක එහෙම නැත්නම් polypeptide chain is not the same the one you find in hemoglobin, right? Or the chain that you find in spider silk, okay? එහෙම නැත්නම් albumin is different to the protein or the polypeptide chain you find in a different protein. It's a unique sequence. It's unique. Then linearly arranged, right? Or beads on a string වගේ නේ මාලයක අර තියෙන beads වගේ. It's linearly arranged. Then it's composed of amino acids, right? The individual components are amino acids. Then how are they connected? By peptide bonds. ඔක්කොම words ටික එන්න ඕන. හරිද? Right. So you can see here in this small section you can see 1, 2, 3. 3 amino acids. Yeah. Connected by peptide bonds. That is a primary structure. Okay? Primary structure. So up to that, fine. Then primary structure is useless. So imagine you have a nice chain. This is useless. This is not a final product. Then okay, our birthday cake hadana then you have connected some amino acids. Then නිකන් අර cake batter එක විතරක් තියෙනවා. Eggs උයි, flour, sugar, තමුණක් දාන්න. baking powder තමුණක් දාන්න. ඔක්කොම ටික දාලා batter එක කරලා තින්නේ. එතන ඉවරද? No. You have to bake the cake and do other things නේ. So this is only the first step. Okay? You just have you only made the sort of basic sort of the precursor. You have to do a lot more things to have a working protein. Right, page number 17. Secondary structure. Secondary structure. Okay. The primary structure of a single polypeptide chain coils and folds. Either it can coil. මොකද coil වෙනවා කියන්නේ මෙහෙම. Yeah. Coil like a spring. Or fold, okay, as a result of intramolecular hydrogen bonds. Intra කියන්නේ මොකක්ද? Intra ගේ opposite එක කවුද? What is the opposite of intra? Inter නේ. International. That means in between nations. නේද? So in between two things. Intra means within the same thing. Okay? Inter කියන්නේ in between, intra කියන්නේ within the same. එතකොට intramolecular කියන්නේ මොනවද? Within the same molecule. Got it? Intramolecular hydrogen bonds, දැන් මම ඉස්සරහට එක underline කරලා තියෙනවා. Between the hydrogen atoms and the oxygen atoms and the hydrogen atoms attached to the nitrogen atoms. Okay? There's a small part missing there. Okay? Oxygen atoms of there's a small printing mistake there. Oxygen atoms of the carboxyl groups. Can you correct it please? Right? oxygen atoms of the carboxyl groups. Then correct karanna. Then bonds, intramolecular hydrogen bonds between oxygen atoms of carboxyl groups. The main carboxyl groups. Oxygen atoms of the carboxyl groups. This is the carboxyl group. Okay. This is the oxygen atom of the carboxyl group. Okay. And the hydrogen atoms of the amino groups kila correct. Hydrogen atoms of the amino groups. Hydrogen atoms of the amino groups. So these are the hydrogen atoms of the amino groups. Right. This is the amino group. I'll read again. Right. Intramolecular hydrogen bonds between the oxygen atoms of the carboxyl groups and the hydrogen atoms of the amino groups. Correct, Correct. Correct. Of the same polypeptide chain. I told you, same chain okay? Same polypeptide chain backbone to form the secondary structure, which is either beta pleated or alpha helical. Either beta pleated or alpha helical. So I have given you the both structures there. Can you see in this helix spring like structure, okay, right? In there, what is this red color atom? Red color ball, that is oxygen. You can see in the previous diagram, I had given it labeled. The red ball is what? Oxygen. Then, what are the hydrogens? Hydrogens are the grey color ones. Grey color ones are the hydrogens. So, can you see between the oxygen and the hydrogens, you have hydrogen bonds forming. So, if I try to draw this, right, alpha helix and beta pleated sheet are the two types of secondary structures you need to know. It's always going to be intramolecular, within the molecule hydrogen bonds. Within the molecule hydrogen bonds. Now imagine you have a carboxyl group like this, right? And it is connected to by a peptide bond. Okay, it is connected by a peptide bond, right, to a nitrogen which has a hydrogen here. Then home it's going to bend. Okay, there's a carbon here, there's a hydrogen here, there's a R group here. Okay, right, there's a R group here, then there's a C double bond O here. Then I'm trying to make a helix, right? There's another peptide bond here, and peptide bond connect there's another nitrogen, hydrogen, and another carbon and a hydrogen. Another R group R2 okay. Then there's another C double bond O. Okay. Then there's another peptide bond. There's another peptide bond, right? A curve in there, right? Just to show you the helical shape. Then there's another nitrogen here, there's a hydrogen here, connected to another carbon. Okay, hydrogen, there's another R group. Okay, then there's another nitrogen, there's another carbon, double bond. Okay, oh man, right? This is the helical shape, right? There's a helical shape, right? Okay, spiral එකක් වගේ. So how are you going to get how is this spiral maintained? There should be something maintaining the spiral. Who is going to maintain the spiral? Hydrogen bonding between who? The oxygen of the carboxyl group නේ. දැන් මෙහෙට තියෙනවා මේක මෙහෙම තව දිගට යනවා spiral එක. Okay. Oxygen of the carboxyl කියන්නේ මෙයා. නේද? Yeah. Oxygen of the carboxyl and it should connect with what? Hydrogen of the amino නේ. Hydrogen of the amino. දැන් මම මේ hydrogen මේ පැත්තෙන් දාන්නද? Then it's easier for me, right? There's a hydrogen here connect to this nitrogen නේ. මම මේ carboxyl එක පොඩ්ඩක් මෙහෙන් දාන්න. එතකොට it's easier to see. Got it? දැන් මෙච්චර details you don't need. Okay? මේවා අඳින්නවත් මලකට එන්නේ නැහැ. Right? I'm just trying to make you understand. So what do you know about hydrogen bonds? This oxygen is slightly negative නේ. electronegativity කතාවක් තියෙනවා. Then this hydrogen is slightly positive නේ. Then between this oxygen and this hydrogen there's a hydrogen bond. Okay, got it? Then you can also have another තව තැනක් you can, I mean, so this is the only place, right? මෙහෙමයි the alpha helix is maintained. How is it? Between the oxygen of the carboxyl group and the hydrogen of the amino group. Got it? There's another structure that you need to know is called the beta pleated sheet. The beta pleated sheet is exactly the same thing, but the amino acids are running like this. The amino acids, sorry, the polypeptide chain is going to sort of be like the pages of a sort of like the pages of a book. So if I show it to you, if you take this page, if I fold it, no, I need another separate piece of paper, right? So the beta pleated sheet is like a piece of paper that you sort of folded into a shape like this. මෙහෙම shape එක. මෙහෙම. This is a beta pleated sheet. Okay, right? So the polypeptide chains are running මෙහෙම, right? මෙහෙම, right? තේරෙනවද? The polypeptide chains are running like the sort of the planes of this paper, right? So if you are looking at it from the front, you are going to see the foldings. Got it? So where do you have the intramolecular bonding? Where are you going to have the intramolecular bonding? Between these sheets, මෙහෙම තියෙන්න පුළුවන්, right? Among these foldings, you are going to have a hydrogen bonding. Okay? Among these foldings, you are going to have hydrogen bonding. That is called the beta pleated sheet. Alpha helix and beta pleated sheet. What are the examples for alpha helix? මොනවද දීලා තියෙන්නේ? Keratin නේ. Keratin දීලා තියෙනව නේද? Where do you have keratin? කොහෙද? Here. Here you have keratin. I think අද උදේ මම එවුනේ the photo එකක් to the group. How the hair follicle is formed. Didn't I send? එවුනේ? Yeah. If you take a hair follicle, what do you mean by a hair follicle? In your scalp, your hair have a root. Root එකක් තියෙනවා. Hair comes out like this. There's a shaft of hair. Gala, there's a hair shaft, right? So this here you have, I mean, this entire thing is a living thing. The hair is a living structure. Okay? මේ කොනට යන්න it is mainly composed of keratin, keratinized cells. මේ හරිය යනකොට it has a shaft and it has inner cells. It's a living thing. දැන් මෙහි root එකක් තියෙනවා. From the root the hair is absorbing nutrients. Okay? But it's mainly අරහෙට යනකොට it's going to be dominated by a protein called keratin. Okay? So keratin has this sort of a spiral structure. Keratin has this sort of a spiral structure. Okay? So what is the fundamental property of hydrogen bonds? Hydrogen bonds වල මොකක්ද තියෙන basic quality? Easily broken. නිකම්ම කැඩෙනවා hydrogen bonds. Right? That's why you can easily manipulate hair. දැන් ඔය සැලූන් වලට ගිහිල්ලා අපි කරන්නේ like hair straighten කරනවා කියලා මොකද කරන්නේ? you apply some heat, then you can change the way the hair behaves. Konde straight කරන්න පුළුවන් පොඩි වෙලාවකට, right? For a short moment you can change the behavior of your hair. That's because you are breaking these hydrogen bonds in order to change the shape, right? Then I think there's like permanent rebonding or yeah, no, there's a technique, right? You can make the hair permanently straight, yeah, but that is damaging to the hair, right? Hair damage, no, never done, yeah. I think when you do this that sort of things, the hair dies, ne? Konde merro ne? So I think the hair falls away until, so you are going to have the effect until the hair falls away, ne? Kesga merno because of the excessive sort of heat and other treatments. Anyway, but if you just iron your hair, that is something temporary. So this sort of a helical structure is going to be affected. Hydrogen bonds එක පොඩ්ඩකට එහෙම වෙනවා. Got it? So that's all you need to know about the secondary structure, right? So highlight alpha helix example is keratin, highlight kana. Alpha helix example keratin, highlight. Online people, any questions? Clear ne? Beta pleated sheet, spider silk, right? So you have seen දැන් මේ මකුළු දැකලා නැත්නම් I have given, okay, right? What do they say? Spiders secrete silk fibers made of structural protein containing beta pleated sheets which allow the spider's web to stretch and recoil. Then you might have seen ne දැන් spiders webs are difficult to break ne ඇදෙනවා නේ හොඳට. A Spider-Man name, it's like stretching, right? That's because you can see it is composed of this sort of a beta pleated sheet. It can stretch, right? Spider silk has this ability to stretch. That's because of its secondary structure. Third one. Any questions? Then let's move on. දැන් අපි නිකන් කේක් එක බේක් කරා වගේ. දැන් පොඩි functional form එකකට ඇවිල්ලා තියෙනවා. නිකන් butter cake එකක් හම්බ වෙනවා. නිකන් ඔය හම්බෙන්නේ. අම්ම හදන්නේ කහ පාට butter cake එකක් ඔන්න හැවිලා තියෙනවා. Right? You are one more step ahead in the sort of process to make a nice cake. හරිද? මේ cake කතාව මොකක්ද කියලා දැන් ආපු right? Right? Okay, we are going to make a functional working protein. හරිද? Okay? Tertiary structure. Usually, the secondary polypeptide chain bends and folds extensively, forming a precise, compact, unique, functional and 3D shape. Okay. There are numbers දාගන්න ඔය වචන ටිකට. Precise 1, compact 2, unique 3, functional 4, and 3D shape resulting from the following interactions between the side chain and the R group. Sorry, side chain or the R group. Okay, මොකද කියන්නේ? So we are finally arriving at a protein that can do work. So if you take keratin and spider silk, those are still structural, they are just structural proteins. Now at the tertiary level, you are going to have working proteins. That is because the secondary chain bends and folds to form a precise, compact, unique, functional, very important word. It has a dedicated function and a 3D, right? 3D shape. Due to the interactions between the side chain or the R group, then mechchara vela we didn't think about the R group, ne? R groups we ignored. Yeah, R groups didn't do anything. Still, it was all, I mean, in the secondary structure, it was the hydrogen bonding was within the backbone, within the backbone. Why is that? Because this oxygen is a part of the backbone. This hydrogen is a part of the backbone. So until now you had backbone interactions only. Now you are going to have side chain interactions, R group interactions at the end. So you can see that clearly. In this diagram you can see in this one. Yeah, you can see I haven't, I mean that diagram haven't drawn the backbone නේ. backbone එක ඇඳලා නැහැ නේද? නිකන් purple color නිකන් tape එකක් වගේ පණුවෙක් වගේ විතරයි තියෙන්නේ backbone එක නේ. Yeah, that is because the backbone is the same. ඇඳලා වැඩක් නැහැ. Yes, backbone is the same repeating structure one after the other. Yeah, carbon amino group, carboxyl group, carbon amino group, carboxyl group, repeating one after the other. So අපි ඒක අඳින්නේ. It's just a backbone. What is important is what is sticking out of it. Side chains are now going to be important. Can you see there are so many types of side chains that is sticking out of it, right? Balamu, then so they say the tertiary structure results from interactions between the side chains. How do the tertiary structure is formed due to interactions between the side chains. Monada may interactions, hydrogen bonds. There's a hydrogen bond given there. Can you see? Label color in the tute. Yeah, there's a hydrogen bond. The OH between the hydrogen of the OH and the double bond oxygen. You can see a hydrogen. The disulfide. Disulfide bond disulfide means between two sulfurs, right? Between two sulfurs, okay. Between two sulfurs, you can have a what we call as a disulfide bond. This is an important bond, okay. So then, inorganic karaddi, you will learn about individual elements. The inorganic lesson I think 4th lesson in another 7 to 8 months, right? When you are doing inorganic, you are going to learn about individual elements, right? Individual elements. So I used to have, for each element, I used to have like a sort of a wallpaper for each element. Oxygen wallpaper, sulfur wallpaper, halogens can chlorine, fluorine, bromine, right? Then nitrogen, okay. The biggest wallpaper was for sulfur. Okay, sulfur forms and involves in so many reactions. Sulfur oxides godak hadanawa, ila acids godak hadanawa, gases godak hadanawa, right? So sulfur is very reactive. It's a reactive element. Okay. So similarly, when you have two sulfurs that are sticking out, you can have a bond called a disulfide bond. Okay, or a disulfide bridge. Then ionic bonds you already know. How do you get an ionic bond? How do you get an ionic bond? Can you see? Between NH3 plus and O minus. Yes. Between NH3 plus and O minus, you are going to have ionic bond. Yeah. Yeah. How do you get an ionic bond between two oppositely charged? Between a positive charge and negative charge species, may have positive, may have negative. So this is a ionic bond. Very strong bond, okay. What is Van der Waals interactions? Van der Waals interactions. What do you mean by Van der Waals? Van der Waals London forces okay. Tama karene the atomic structure. You are the atomic structure. Not yet. Chemistry class okay. Then what we are talking about here are called intermolecular bonds. Bonds between molecules. Bonds between molecules. There are, almost all the bonds are given here. Hydrogen is a type of intermolecular bond. Then disulfide bonds can form. Okay. However, there's a type of bond called Van der Waals, or you can call them hydrophobic. Api Van der Waals consider karanne, we only consider Van der Waals interactions when there are no other significant bonds. Okay. Usually in normal scenarios, your Van der Waals bonds are negligible. Api ganan ganne. Okay. Usually Van der Waals bonds hadenne. We say between any molecules, any two molecules, you have Van der Waals bonds. Okay. Between any two molecules, you have Van der Waals bonds, but we don't consider them because there can be other stronger bonds, such as what? Hydrogen bonds තියෙන්න පුළුවන්, polar bonds තියෙන්න පුළුවන්, right? You only consider Van der Waals when you don't have any other bonds. දැන් උතන පේනව නේද? CH කියලා කෙනෙක් ඉන්නවා, මෙතන CH3 කියලා එකක් ඉන්නවා. There's another CH3. Then on this side there's a CH and there's a CH3. And මෙතනත් තියෙනවා CH3 එක. පේනව නේද? There's a pink color box පේනව නේද? So we say between these two within Natare, we say there's a van der Waal interaction. It's very negligible force. Van der Waal interactions occur between non-polar groups. Polar කියන්නේ දැන් පේනවද? මෙතන එක කිසිම charge එකක් නේ. There's no charge මේ වගේ. There's no uneven charge distribution, nothing. Okay? Very weak interaction, but it's still there. And the other thing you need to know, it's a repulsion. Van der Waal කියලා කියන්නේ attraction එකක් නෙවෙයි. හරිද? Van der Waal කියලා කියන්නේ repulsion එක. Attraction එක නෙවෙයි. Okay? Interactions කියලා අපි කියන්නේ. Interactions can be attractions or repulsions. නේද? Did I ever say these are all attractions? No. තියෙන්නේ මොනවද? Interactions. පේනවද නේද? So van der Waal or hydrophobic, they are repulsions. ඒගොල්ලෝ කැමති නැහැ. They repel. Okay? Whatever, it's an interaction. So it's going to contribute to the final shape. තේරුණාද? Right. So what are the examples? Most of the enzymes, myoglobin and albumin. Right, myoglobin and albumin. දැන් මේ පැත්තේ ඔයාලට දීලා තියෙනවා this is myoglobin. label එකක් දාන්න myoglobin. Okay, මේ පේනවනේ මේ කොනේ ඉන්නේ මේ right myoglobin. Put a label that is myoglobin. Okay, myoglobin. It යටින් අපි ලියමු. Right, underneath that diagram of myoglobin, let's write down myoglobin stores oxygen. Myoglobin, okay, ඒක මම කතා කරන්නේ myoglobin ගැන. Myoglobin stores oxygen in muscles. Myoglobin stores oxygen in muscles. Myoglobin stores oxygen in muscles. Okay, online people writing, right? Myoglobin stores oxygen in muscles. Right? So that is a functional protein. Myoglobin. Can you see that myoglobin it's mainly composed of helixes, ne? Alpha helixes වල ඉන්නේ පේනව නේද? spirals වගේ තියෙනවා. Yeah? And all those helixes are clumped together by what? These interactions. One of the hydrogen bonds, disulfide වෙන්න පුළුවන්, ionic, whatever. Tertiary level එකට එනකොට you are going to have sort of them grouping, I mean becoming a compact structure. Ne? That's how we describe it. Precise, compact, unique. How many describe kare? Got it? Got it? Albumin we'll discuss later. Albumin is a protein found in your sort of blood, right? Blood protein එකක්, circulating protein එකක්. හරිද? So these are the things you need to know about the tertiary structure, right? Now the tertiary structure is actually a final sort of a cake, right? You can, I mean, you can put it in the shop or you can right, as a birthday cake. You can go to the quaternary level to make like multiple tattu තියෙන cakes තියෙන, right? You can make it more complicated, right? At the tertiary level you have a completed product, right? You have a completed product. නේ අර තනි තට්ටුවේ cakes තියෙන. It's like that, right? But if you want to make it more elaborate, okay, you can go up to the quaternary level, right? Quaternary structure. What do you need to know about the quaternary structure, right? Is that to make a quaternary structure you need 2 or more polypeptide chains. At least දෙකක්වත් ඕන. Okay? To make a quaternary structure you need 2 or more polypeptide chains. So until this point you had only one polypeptide chain. එකයිනේ හිටියේ. Right? එකෙන් තමයි මේ ඔක්කොම ගෙව ගැහුවේ. Right? Until now you only had one polypeptide chain. From this point onwards at the quaternary level you are going to have 2 or more, at least දෙකක්වත් ඕන. Right? 2 or more polypeptide chains. The best example I can tell you is, I mean one of the commonest examples is hemoglobin. Hemoglobin. අපි කියවලා බලමු. Page number 17 යටම, okay? Aggregation of two or more polypeptide chains involved in the formation of one functional protein. Okay. Two or more polypeptide chains. Highlight that. You have two or more polypeptide chains. Separate chains are called protein subunits. Separate chains are called protein subunits, which are held together by inter and intramolecular interactions. Okay. Right. Let's take a look at our own very close to our life hemoglobin. Very close to our sort of physiology hemoglobin. Hemoglobin molecule has 4 globin proteins. They make a globin protein. Okay, it has 4 globin proteins. This is the other one. Okay, hatarak. Okay, it has 4 globin proteins, 1 and 2, and this is the other one. Okay, it can have so many. Then parent secondary structure can be alpha helical or beta pleated. They join by what? Other interactions to become the tertiary structure. Several tertiary structures get together to form the quaternary structure. So that's so 4 such structures get together. Up here name deno alpha 1, alpha 2. The blue color ones are beta 1, beta 1 and beta 2. So hemoglobin has how many polypeptide chains? Individual 4. Other than this, hemoglobin has another special feature. It is what we call as a metalloprotein. It's a metalloprotein. Have we had a metalloprotein here? It is one of the places in your body that we have iron. Fe2 plus. Hemoglobin has an iron atom or a iron, right? Fe2 plus. There's a one in each of the subunits. Okay. Then one mega. Yeah. So this presence of this metal ion gives the hemoglobin an ability to carry or to bond with oxygen. So this oxygen will be connected to each of these, right? Each of these subunits has the ability to transport one oxygen molecule. What is it? Each of these subunits has the ability to transport one oxygen molecule. So what is the transport capacity? අපි දැන් ලොරියක උනත් කියවනේ කිලෝමීටර පටවන්න පුළුවන් කියලා නේ. So what is the transport capacity of oxygen by a single hemoglobin? අපි සාමාන්‍යයෙන් we indicate hemoglobin by Hb. Okay, Hb plus oxygen. How many oxygens? Kiya connect කරන්න බලන්න. 4. Yeah, clear, right? Following me, right? So one hemoglobin can connect with 4 oxygens. So what is the balance equation? අපි ලියනවා මෙහෙම HbO8. Yeah, oxygen atoms 8ක් නේ. At the end of the day it's 8 oxygen atoms නේ. 4 oxygen molecules, so 8 oxygen atoms can be transported by one hemoglobin. Right? This is something you need to know. The oxygen can transport the ability of hemoglobin. Okay. So if we just go into the resource book diagram, hemoglobin, I have given you the alpha, beta, heme ion, kalu පාටට පේනව නේද? Page number 17 atom online paper. Right side bottom diagram. Yeah. So where is hemoglobin present? Where do you find hemoglobin? Inside red blood cells, right? if you take a red blood cell has a very unique shape. The udin baluwa mehem pene, I am not very good at drawing. Abi red blood cell එක පැත්තෙන් බැලුවාම it's like this නේ. අපි මේකට මොන වගේ shape එකක් කියලාද කියන්නේ? මේකට අපි lens જાතියකුත් තියෙනවානේ bicon cave නේ. Bicon shape එක. So I don't know the mathematics behind this. Then මේ shape එකේ surface area is greater than just a ball or a sphere or even මෙහෙම shape එකකටත් වඩා or even than this has greater surface area. Okay, this shape has greater surface area. දැන් අපි ලොරියක් ගත්තොත් if you want to quickly load and unload, what do you need to have? There are වැලි පටවන ලොරි වල තුන් පැත්තක් අරින්න පුළුවන් නේද? You can open three sides. Have you seen? Yes, that means it's easier to load and unload. Ne? The greater surface area you have, it is easier to load and unload things. So in the red blood cell, what do you have? Hemoglobin. Hemoglobin transports what? Oxygen. So when you have more surface area, it is easier to load oxygen and also unload oxygen. Ne? It's a very scientific shape. Got it? Red blood cells. Okay, red blood cells can other features you will learn. Then red blood cell there is no nucleus. There is no mitochondria. Mukut nahi. It's like a driverless lorry. You know, its only purpose is to transport things. No controller, nothing. Right? Because why? To save the space. Nucleus nahi, mitochondria nahi, nothing. Right? It's just a lorry. Right? China there are delivery vans that are sort of automated ones. Right? There's no one here. It is just designed to transport oxygen and also some carbon dioxide. Okay, that is hemoglobin. Anith ekena balanna collagen. This is collagen I have given here, bottom of the page. This is collagen. Okay, collagen, can you see how many subunits? How many protein subunits does collagen have? Balla kiya? Yeah, kiyatena. There's a green one, there's a blue one and a red one. Three, ne? Three. Collagen has 3 subunits. Okay? Collagen has 3 subunits. Where do you find collagen in your body? Body ke koi the collagen thi. Where do you have collagen? I told you in underneath your skin you have. So if you take like a bone, then this is the humerus, right? A forearm bone. So for the forearm bone is connected to another bone. I'm sorry, this is your arm bone. It is connected to your forearm like this. Then here, what do you have? You have your biceps muscle. You have your biceps muscle. Yeah. So if imagine you want to bend your arm like this, how is it going to do that? This muscle is connected to the forearm by a tendon. Yeah, this tendon is mainly composed of collagen. We call those type of tissues as connective tissues. Okay, they connect two tissues together. They connect muscle to the bone. Right? So in such places you have a lot of collagen, right? So main diagram mega, right? Can you label it as collagen? Okay, collagen. Right? Here I think අපි පොඩි sentence එකක් ලියමු underneath the collagen. Okay, collagen provides strength to dense connective tissue such as tendons and ligaments. Such as tendons and ligaments. Collagen provides strength to dense connective tissue such as tendons and ligaments. got it? Collagen. මෙයාව ලේබල් කරන්න දෙයක් නැහැ නේ. It's given as hemoglobin නේ. හරිද? එයාව hemoglobin විදියට දීලා තියෙනවානේ. උඩින් ලියන්න මෙයා this is label this one as red blood cells. Okay? RBC, red blood cells. Okay. RBC or your red blood cells. So at this stage, things like this. Then මෙයාව ගත්තාම this is like a multi-layered cake නේ. තට්ටු ගානක cake එකක් වගේ because you have combined several polypeptide chains. ඒ අසට metal ion එකකුත් දාලා like it's like අර birthday එකට already පත්තු වෙන ඒ වගේ you have made this cake very elaborate, right? It's a mix of metal and protein that is able to do a very complicated function such as transporting oxygen. Then it is not just loading and unloading oxygen. Hemoglobin has the ability to detect where in the body you need oxygen. Okay, so it has the ability to sense. So for example, hemoglobin molecule in your lungs, he detects that okay මෙතන මට oxygen ගොඩක් තියෙනවා. So what I need to load oxygen right now. In the lungs, hemoglobin load oxygen. Okay, so it it circulates through your blood and goes to a muscle. So then duano. So it reaches a working muscle. So in that place, the hemoglobin detects that okay, in here the oxygen concentration is less. So what do I need to do here? I need to release the oxygen. Okay, it's like that. So it's a working protein. So because of this metal ion, it is able to combine with oxygen. Okay, you will learn when you are doing the circulatory system on 2027 I start කරන්න යන්නේ that lesson. Hemoglobin has the ability to transport carbon dioxide as well. Okay, that carbon dioxide joins to parts of the hemoglobin molecule other than this metal. Iron එකට නෙමෙයි ඒගොල්ලෝ join වෙන්නේ. They join to sort of these protein parts, right? They connect to these sort of proteinous components. So the hemoglobin molecule can also transport carbon dioxide as well, right? Not only oxygen. On the loriac, okay, right? Very good. With evolution, it has become a very efficient molecule. Okay. Metal, right? Fe is found mainly in your red blood cells. You have 10 million, more than 10 to 15 million red blood cells at a given moment. So there you have a lot of hemoglobin, iron. X-Men එකේ තියෙනනේ magneto කියලා ඉන්නේ. There's someone who can control magnetic things. දැන් බලලා තියෙනවා. Yeah? X-Men බලලානේ. He sort of absorbs all the metal from sort of the iron from someone, right? Like a prison guard connecting, he absorbs all the iron that is found in hemoglobin using his powers. Okay, right? There's something to learn. Okay, right? I'll send you the clip. Right? He escapes from the prison using the metal in the prison guard's body. Okay, he's like kept in a sort of a full plastic environment because he can control the metal and escape. Baladina, yeah, okay. මේ ටික කාලේ පොඩි movies ටිකක් බලන්න, right? They have some things to learn. Okay, right? So that's because if you take all the red blood cells and all the hemoglobin inside them, you have a significant amount of iron. I think close to 1 gram or something, right? Very substantial amount, right? Right. Clear? So we have completed all the levels of protein structure, right? ඔය බලමු question එකක් ගන්න පුළුවන්ද කියලා. නැත්නම් දොයිය ගන්නවනේ. Right. Not yet. දැන්ම ගන්න බෑ. No, දැන්ම ගන්න බෑ. Right, let's move on. හරිද? Go to page number 18. Denaturation of proteins. This is a very important concept, right? So what is the property in this world, right? Earth එකේ තියෙන principle එකක් තමයි, beautiful things are destroyed easily, ne? Complicated things are easy to destroy. Yeah? When things are more complicated, or more complex, yeah, they get easily changed. Ne? Simple things don't change much. So things like these very elaborate proteins, they are held together by weak interactions, hydrogen bonds, van der Waals, yeah, they are weak, therefore they are easy to break, right? That's why proteins can be easily destroyed or changed. You can destroy from the original structure. If they are changing, that is called denaturation. Highlight, this is the definition. Denaturation of proteins, the loss of specific chemical 3D shape due to the alteration of weak chemical bonds and interactions. Specific chemical 3D shape, very important set of words. I'm sorry, මේ කලින් page එකේ මෙන්න මේ reaction එක ලියා ගන්න. Underneath the hemoglobin, okay. Online people, on the previous page, underneath the hemoglobin molecule, write down this reaction. Okay. And it යටින් ලියන්න මෙහෙම deoxy hemoglobin. ඒ කියන්නේ hemoglobin without oxygen is called deoxyhemoglobin. This is called oxyhemoglobin. Deoxyhemoglobin plus oxygen forms oxyhemoglobin. Sorry. Questions වල අහන්න පුළුවන්. How many oxygen molecules can be transported? What is the answer? 4. How many atoms? 8. I mean, you will learn that oxygen O2 is connected by 2 bonds. Ne? Oxygen atoms are connected by 2, I mean, it's a double bonded molecule. Ne? Okay? It's very difficult to break that. Okay, clear? Then what we have to learn about is denaturation. Denaturation. So what is meant by denaturation is, it's not just it's not the complete destruction. Just one level of protein structure going back. Then imagine you were in the tertiary level, you were sort of demoted to secondary level. That is a denaturation, right? කොහොම හරි from your proper structure, if you were if your structure was altered, that is a denaturation. Okay, so ඔතන මම example එකක් දීලා තියෙනවා. I have given there's a process of frying an egg. Can you see? I have given you a diagram, right? Can you see? Albumin. So ovalbumin is the protein found in egg white. Okay, egg white එකේ තියෙනනේ proteins දන්නවනේ. Yeah. So next week for the experiment to identify proteins, we are going to use egg white as well. Egg white proteins. Okay. The පේනවනේ when you apply heat, විතරක් fry කරලා තියෙනවනේ. When you apply heat, what's going to happen? It's going to gradually become white colored, ne? And it's sort of liquid nature becomes sort of jelly like and solid, ne? Yeah, so that is because of what you are applying, because of the heat you are applying, the protein is undergoing some changes. There is a form of denaturation. Got it? So agents affecting denaturation. Agents of denaturation. Okay, there's space to write down 4. Got it? You need to be able to precisely write down these 4. Okay. 1, high temperature, high temperature and high energy radiation, high temperature and high energy radiation. 1. 2. Strong acids, alkali and high concentration of salts. 3. Heavy metals. 4. Organic solvents and detergents. Organic solvents and detergents. Okay. Agents of denaturation. You can see, of course, high temperature and high energy radiation is able to alter bonds, right? When you apply a lot of energy, so in the form of high temperature, or it can be radiation as well. Radiation, you will learn about 3 types of radiation. There is alpha particles, beta, and then also there is electromagnetic, right? So there can be particulate radiation, then sort of energy radiations as well. Okay, different kinds of radiations are there. All those are, what do you mean by radiation and also this high temperature? They are ways of sort of transferring energy. You are applying a high temperature means you are transferring heat energy, you are giving energy. Yeah, in radiation also you are giving energy. So when you provide energy, තියෙන bonds can be altered or broken down, new bonds can be formed, whatever. Okay. The high energy radiation, so you must have seen, you must have heard, right, sort of nuclear radiation. What is the sort of nuclear radiation can have sort of short term and long term impacts, right? So immediately then, for example, then Chernobyl wage TV show එකක් බලලා තියෙනවද about the Chernobyl nuclear disaster? ඒවත් බලන්න. Okay, only 6 episodes. Okay, watch. Okay, right, homework. So anyway, in that sort of a nuclear exposure, immediately ළඟම හිටපු set එක මැරෙනවා. That is because of the short term effects of radiation, right? All the proteins in their body directly exposed, such as their skin, such as their blood vessel walls, such as their, I mean, every sort of skin cell in their body is going to be affected by the high energy radiation. Then if imagine they got like a secondary dose, then the radiation particles travel by wind in the water, right? Imagine they were exposed like indirectly. That is going to affect not their proteins but their DNA. So that's what we are going to study later today, right? So that high energy radiation can have short term effects on the proteins due to direct contact, long term on the DNA. Then DNA effect it can cause sort of negative effects in the next generation. Okay, if you are sort of reproducing, sort of the reproductive cells got the radiation, there can be defects, sort of birth defects lamai hambeno in the next generation. That's what happened to Japan after these nuclear bombs, right? So immediately bomb එකේ blast radius එක හිටපු set එක wash වෙලා ගියා. Yes, but people who got exposed to the radiation, their children or the subsequent generations had got sort of defects, sort of extra limbs, extra digits, and mental retardation, cancer. So many negative effects were there, right? That is because of the DNA affection. This is the short term effect, that is due to the high energy radiation, proteins can be destroyed. Okay, then in that in that TV show, I think it's a bit exaggerated. They say to this, there were firefighters who were going to this nuclear power plant, right? It was a fire, no, so firefighters were the first people there. These firefighters, so they got radiation burns. So when they were taken to the hospital, they say, so they were in extreme pain. So they say you can't administer any painkillers because their blood vessels are all open. The blood vessel walls have been destroyed, so the blood is not flowing in the vessels. It is just in the body. The blood vessel wall proteins have been destroyed. So when you give a sort of painkiller injection, it doesn't work because it's not flowing, right? You can't even give painkillers for their pain, got it? So maybe a bit exaggerated, but it can happen, right? Because of the protein damage caused due to the high energy radiation, right? Then second one, this word is very important. Had the strong window, had strong acids and alkali vitarai usually going to have an effect, because I told you, I mean, you know, your amino acids are what? Amino acids have amphoteric nature. Ne? So if you take an amino acid, it has this COH group, and on this side you have the amino group. Ne? So මෙයාට පුළුවන් අපි කියනවා this amino acid can act as a buffer. Buffer කියන්නේ මොකක්ද? Buffer is something that resists change, right? This is a acid buffer or a acid base buffer. දැන් මෙයාට අපි හිතමු you give this guy an acid. අපි acid එකක් කියලා H+ එකක් දැම්ම කියලා කිව්වොත්. This H+ is going to join with this amino group and you are going to get NH3+ කියලා group එකක්. Absorb කරගන්නවා this hydrogen. හරි? Imagine you gave this an OH-, මෙන්න මෙතනින් hydrogen එකක් ඇවිල්ලා combine with this OH- to get a water. Neutralized. Okay? So amino acids are able to resist acids and bases up to a certain level. හැබැයි නිකන් ඩෝන් ගාලා concentrated acid එකක් දැම්මොත් ඔක්කොම ටික destroy වෙලා යනවා. Got it? So amino acids are buffers but not for strong ඒවට කරන්න දෙයක් නෑ. Got it? Clear? In inorganic chemistry, when you are doing buffers, I clearly remember we chemistry class එකෙත් you will draw this amino acid. Okay? Amino acid එක ඇඳලා acid එකක් දැම්මම මොකද වෙන්නේ? base එකක් දැම්මම මොකද වෙන්නේ කියලා you will draw. Okay? I exactly remember the day, right? Yeah. That we drew this, right? Amino acid act as buffers, but only for weak acids and bases. තේරුණාද? ලොකු තඩි acid එකක් දැම්ම කියලා high nitric acid දැම්මොත් එහෙම ඉතින් ඉවරයි. Yeah? So strong acids, bases එහෙමයි. Salts වලදී වෙන්නේ salts have a sort of a dehydration effect. Okay? Salts can absorb water. So that is going to alter all these other bonds. Hydrogen arome okkom effect. Heavy metals. What is the effect of heavy metals? What do you think? Heavy metals වලින් මොකද වෙන්නේ? Heavy metals are what we call as catalysts. What do you mean by a catalyst? Catalyst is something that can promote a reaction. ඔලවලට තිබ්බ නේද? Nickel as a catalyst, iron as a catalyst. Yeah? තව මොනවද? lead catalyst තිබ්බ නේද? මොන හරි nitric acid හදනකොට මොන හරි තිබ්බනේ lead. I can't remember now. Right? So heavy metals act as catalysts. Then උන් ඇවිල්ලා they are going to promote other reactions in the protein. Okay? So that is also going to affect the structure of the protein. Got it? Organic solvents and detergents. Right? Organic solvents are designed to dissolve organic molecules. Dissolve වෙනවා කියලා කියන්නේ මොකක්ද? They are surrounded by whatever the molecule of the solvent. Right? What is the most common place that you use these organic solvents and detergents? As disinfectant. Dettol, phenol, or මේව කියලා දාලා හොදන්න. Yeah? So in the hospital, අපි දැන් සාමාන්‍ය washroom එකට ගියාම ඔහොම ගොඩ ගහලා තින්නේ in the sort of the staff washroom ඔක්කොම phenol අර මේවා තමයි ගොඩ ගහලා තියෙන්නේ. Ophthaldehyde or මේවා sort of hypochloric acid ඔක්කොම detergents ටික තමයි තියෙන්නේ. Because in a hospital setting we use a lot of detergents, right? And disinfectants. Yeah? So they are designed to destroy proteins. What proteins? Bacterial proteins, ne? Viral proteins. ඕවනේ destroy කරන්න in a hospital. So that's why organic solvents and detergents are able to destroy proteins, denature proteins. Got it? ඔය ටික තමයි agents of denaturation. තේරුණා? Any questions? Any questions? චුටි question එකක් තියෙනවා page number 22 එක MCQ එකක් තියෙනවා page number 22 MCQs වල can you see page number 22 වල යටම තියෙන්නේ proteins කියලා question එක proteins statement number 1 form the secondary structure due to disulfide bonds are made up of 26 amino acids composed of carbon hydrogen oxygen nitrogen අනේ question එක ගන්න බලන්න and say tell me the answer. Online people send me the answer as well. Yeah, what's the answer? What's the answer? Proteins form the secondary structure due to disulfide bonds? No. ඉස්සරහින් ලියා ගන්න. Right? Secondary structure is formed due to is formed due to what? Intramolecular hydrogen bonds. Write down a small review. Secondary structure due to intra right intramolecular H bonds. Right? හොඳට intra, okay, made up of 26 different only කපලා හදන්න එක 20 නේ 20 different amino acids are composed of carbon hydrogen oxygen nitrogen sulfur and phosphorus. Phosphorus is not very common නේද? Okay, so you can cut phosphorus. sulfur වෙනකන් තියෙනවා සාමාන්‍යයෙන්. Okay, sulfur is a very common element in proteins, not phosphorus. Okay, would not be denatured by detergents. List එකේම තියෙනවානේ. That is wrong. Contribute to transport of materials. Hemoglobin, got it? Hemoglobin, there are other things within, right? Serum albumin, right? We'll talk about the functions very soon. Clear? Hi, I think we are going to move on to the sort of the most important thing. Now, we protein එක හැදුවට we need to have an extensive idea what are the different functions of proteins. Okay, so for that මෙන්න මේක තමයි your guide, page number 18. Okay, I have given it in full color because otherwise no use, right. So, using this diagrams, I want you to have a visual idea, okay, this is the list of things that a protein can do. This is the list of functions that can be performed by a protein. Okay. So, I am going to relate it to these diagrams to the functions that are given in your resource. Okay. So, that is how we are going to have a complete list of protein functions with the diagram. Okay, complete list of protein functions with the diagram. Okay. Right. So, there is some differences between those diagrams and the way they are given in the resource. So, I will try to go according to the resource book order, right. So, the first protein function that is given in your resource is එක පිළිවෙලට තියෙනවානේ. හරි. Protein functions. හරිද? First one, catalytic proteins. Catalytic, or you can say enzymatic protein functions. First one, catalytic or enzymatic. Then what is the function? So, I am going to write in 3 colors. Okay. Catalyze, biochemical reactions, that is the function of catalytic or enzymatic proteins. The examples are pepsine and amylase. Pepsine and amylase. So, check your first box, enzymatic proteins. එතන ලියා ගන්න as the function. එතන function කියන එක it's not exactly what I want you to have, right. First box, function catalyze biochemical reactions. Write down there. Here in this first box, function is catalyze biochemical reactions. Examples, so they have given digestive enzymes කියලා. These are the names of the digestive enzymes, pepsine and amylase. First function, catalytic or enzymatic proteins. That is it. So, there is a green color 2 molecules that are coming and connecting with the purple color enzyme, and they are leaving as 2 molecules. It's like a divorce එකක් වගේ නේ. දෙන්න එකට එනවා වෙන්නම යනවා නේ. Right. That's what enzymes can do. They can break proteins or they can make protein. Sorry. They can break bonds or make bonds. Got it? Any idea නිකන් මොකක් වගේද ඔය එන්නේ නිකන් ඔය green and the 2 green color molecules, what can they be? Any idea? නිකන් it's like glucose and නිකන් fructose වගේ නේද ඒක නිකන්. නිකන් එහෙම හිතෙන්නේ නැද්ද? Fructose is like පොඩක් pentagonal වගේ නේද? It is probably a sucrose molecule may end. Okay, may end sucrose එක කියන්නේ glucose and fructose. Can you see? Right, just for fun. Okay, so catalytic proteins. Next function අපි යමු structural කියන එකට. Okay, structural is given at the right hand bottom. Okay, structural proteins, second function, structural. According to the protein, they have given separate functions. So, they have given as examples keratin and keratin and collagen. Keratin and collagen are the examples. Keratin prevent desiccation. I will explain, okay. Keratin prevent desiccation. Collagen provides strength and support. Provides strength and support. Okay. එතන ලියා ගන්න. So structural proteins කියන තැන write down keratin prevent desiccation. Collagen provides strength and support. මෙන්න මේ box එකේ. Okay. So there's collagen is given again. එතන දීලා තියෙනවා collagen. Got it? Online people? Then, what do you mean by desiccation? Prevent desiccation. Desiccation is another word for water loss. Okay. So, how does keratin prevent water loss? Imagine you were a hairy animal, and you are like a monkey, you are going to be covered with hair. Ne? So, the keratin, I mean hair is composed of keratin. Ne? Yeah. So, because of that, the hair is acting as a sort of a layer above over your skin, so that prevents water loss. Even in our skin is composed of this sort of our skin is composed of multiple layers. Do you know this? Ape skin ekata there are multiple layers of cells kila, right? Yatama thiyena cells are more sort of white. Udata yanne yanne the cells become more and more flat. The cells become more and more flat. Okay, the cells become flatter and flatter. At the skin surface, the cells are like very thin plates, okay, and they are dead. Our skin surface cells are dead, okay, and inside them they are filled with keratin. Ape surface එකේ තියෙන skin cells, they are dead and filled with keratin. Okay, you can see this very clearly in places where you have a much thicker skin. කොහෙද? Palms, and where else? Soles of feet. You have thick skin. Yeah. When you sort of, you can remove that skin. Sort of white color layer. That is because it's dead skin filled with keratin. Okay. So because of that keratinized cells, your skin surface is a good sort of a waterproof barrier, and also it is good. I mean, it is resistant to sort of abrasion. වැඩක් කරා කියලා එහෙම එකපාරටම ලේන්න ගන්න නැහැ නේ palm එකේ. Yeah, it's able to withstand friction. So that's why we say keratin prevents desiccation. In the hair or in the skin, keratin prevents desiccation. Got it? Alright. Next, storage. Okay, next function, storage. You can see හොයාගන්න පුළුවන් නේ මෙතනදී storage proteins. You have again two examples for storage protein. You have ovalbumin, which is the storage protein in egg, right? Which is the storage protein in egg. Then we have casein, which is the storage protein in milk. Okay, casein is the storage protein in milk. Okay, storage protein in milk. එතන ලියා ගන්න. Casein දෙක දීලා තියෙනවා නේ. Casein is the protein of milk. Ovalbumin is the protein of egg white. කියලා දීලා තියෙනවා නේ එතන. පේනව නේද? So එහෙනම් ලියන්න ඕනෙත් නැහැ. Okay. Clear? Then transport proteins. Next function transport protein. कोई diagram එකේ තියෙන එක. Transport proteins ඔතන දීලා තියෙන්නේ it's a different type of transport. Okay, so in this box you have given transport proteins දීලා තියෙනවා නේද? Right? So transport can be two types, right? In your resource they have given a different type of transport, but අපි දෙකම ඉගෙන ගමු. Right? Function number 4, transport. Okay, what are the examples? Hemoglobin, what does hemoglobin do? Transport oxygen and right? Transport oxygen and carbon dioxide. Then this is albumin. Albumin is a protein when you find it in the liquid part of blood. Okay, your liquid part of blood is called plasma, but in plasma we can also call it as serum. Okay, so the albumin you can find in serum is called serum albumin. Serum albumin. Serum albumin has a function of what? Transporting fatty acids. Transport fatty acids. Can someone tell me, why do we need a protein to transport fats? Protein to transport a fat. Special. Why do we need a protein to transport fat? In blood. Blood is mainly water. Blood is mainly water. Liquid part of blood is water. So, to transport fatty acids, why are we using a protein? Imagine you have like a bowl of fat, lipids, that needs to be transported in the blood. It can't be done, because why? Fat doesn't dissolve in water. Fat doesn't dissolve in water. So, if something doesn't dissolve in water, can you transport it? No. Yeah, it doesn't travel with blood. So, how can you transport it? You are going to put it into a sort of a package. You are going to package this lipids in a sort of a capsule of protein. Protein petti එක දාලා යවන delivery එක වගේ. Yeah, something you can't send by itself, you are going to package it into a protein box and send it through blood. Got it? So, these are the green color, these are the albumin molecules. In the center, you are going to have lipids. Okay? අපි මේවට කියනවා lipoproteins. Okay? We call these lipoproteins. හරිද? නේ අම්මගේ හරි කාගේ ආච්චිගේ හරි lipid profile කියලා කරනවා දන්නවද? There's a blood test called lipid profile. LDL HDL කියලා දැකලා තියෙනවද? ඒකේ levels ගහලා තියෙනවා. Online? Yeah. So this is how you travel, this is how you transport fats. So ඔයගොල්ලෝ blood sample එකක් අරන් if you test for fats, this is what you are going to detect. This is what you are going to detect. The proteins with the lipid inside, these are called lipoproteins. Okay? These are called lipoproteins. අපි ඉගෙන ගන්නවා ඉස්සරහට. Okay? Just telling you lipoproteins. Okay? හරි. මෙතින් අපි මේ කියමු බලන්න මෙතන මේ transport proteins කියන තැන තියෙන the box. Hemoglobin, the iron containing protein of vertebrate blood, transport oxygen from lungs to other parts of the body. Other proteins transport molecules across membranes. Okay, so very soon we are going to study the structure of the plasma membrane or the cell membrane. Okay, we are going to study the structure of the plasma membrane. So you are going to see in the plasma membrane you have protein molecules that act as sort of gatekeepers. Now highway එකේ තියෙන toll gate එක. yeah ඕකෙන්නේ 3 wheeleruයි බයිකුයි ට්‍රැක්ටරුයි ඔක්කොම දාන එක නවත්තන්න. It's like that. In your cell membrane you have specialized proteins that are regulating the movement. Okay, ඒගොල්ලෝ transport proteins තමයි. Got it? Anyway, අපිට මෙතන දීලා තියෙන in your resource they have given only two types of transporters in blood, hemoglobin and serum albumin. Hari, then proteins can act as function number 5 hormones. Proteins can act as hormones. Okay, examples insulin and glucagon. Examples insulin දන්නවා නේද from O levels insulin ගේ sort of the opposite එක කරන එක්කෙනා insulin කරන එකේ අනිත් පැත්ත කරන එක්කෙනා කවුද glucagon නේ. What do they do? Function regulate blood glucose level නේ. Regulate blood glucose level. පේනවනේ දැන් hormonal proteins කියලා there's a box නේ. හොයාගත්ත නේ. පේනවනේ the high blood sugar තියෙනකොට insulin is secreted and that reduces the amount of glucose in the blood. Okay. අනිත් පැත්ත වෙන්නේ මොකක්ද? When you have low blood glucose, which hormone is released? Glucagon. Glucagon is going to increase the blood glucose. ඒ කොහොමද එහෙම කරන්නේ? Glucose magically මවනවද නෝ? How does glucagon increase glucose? By breaking down storage forms of glucose. Right? What is the main storage form of glucose in our body? Glycogen. Glycogen stored where? Liver, muscles. Yeah. When you have glucagon high levels, the stored glycogens are going to be broken down. තව ටික වෙලාවක් කන්න නැතුව හිටියොත් glucose අඩු වුණොත් the fats are going to be also broken down. That is also under the effect of glucagon. Okay. ඕනනේ තව කතා කරන්න පුළුවන් when you have insulin deficiency, what is it? That is diabetes. නිකම්ම diabetes mellitus. Okay. So ඔක පටන් ගත්තොත් ඔයගලන්ට break එකක් ගන්න වෙන්නේ. Okay, right. We'll quickly finish the functions. We'll talk about those in detail later. Hari. නිකන් ජොලියට. හරි. අපි how do you take insulin into your body? The insulin inject කරන අය දැකලා තියෙනවනේ diabetic people. How do you take insulin into your body? Injection, no? Insulin injection. Why do we need to inject insulin? ඇයි අපි කියන්නේ බොන්න බැරි insulin? ඇයි insulin බොන්න බැරි? Because it's a protein. If you eat a protein මොකද digest වෙනවනේ. බඩේ high acid තියෙනවා. Yeah. In any other නිකන් වෙන මොන හරි කෑම කෑවා වගේ තමයි insulin ටික බිව්වොත් it's going to be digested in your body. එයින් තමයි you have to inject it. Got it? From the things you know, you can sort of make sense of the things happening. Bio කියන්නේ එහෙම නිකන් කටපාඩම්ම subject එකකුත් නෙවෙයි, right? හරි ඊළඟ function එක contractile proteins. Contractile motor proteins 6. Contractile / motor proteins. Example myosin and actin. Myosin and actin is it given as contraction of muscle fibers? Okay. Contraction of muscle fibers. Myosin and actin, contraction of muscle fibers. left side bottom box එක නේ මේක පේනව නේද myosin and actin they separately දීලා තියෙනවා what sort of a muscle is this අපි මේකට මොන වගේ muscle එකක් කියලාද කියන්නේ මේ ඉරි තියෙන muscle ඕලෙවල් වලට තිබ්බ නේද striated muscle right so usually myosin and actin can be found in other muscles smooth muscles වලත් තියෙනවා ඔතන දීලා තියෙන්නේ it's a striated muscle it's this is a skeletal muscle ඇත්තටම දීලා තියෙන්නේ okay right in this diagram it's a skeletal muscle tissue okay you can see myosin is the purple color actin is the orange ne ඔගොල්ලෝ syllabus එකේ you have the entire theory of how these myosin and actin work together to make the muscle contract okay I won't talk about it it's called the sliding filament theory okay finally defensive proteins okay defensive defensive proteins example immunoglobulins immunoglobulin function neutralize foreign substances neutralize foreign substances foreign bodies क्यों neutralize foreign bodies so immunoglobulins you can see in the diagram I have given you can see අපි ඔයාලට කියනවා antibodies කියලාත් කියනවා okay immunoglobulins are also called as antibodies okay right you can see yeah immunoglobulins or antibodies are sort of pieces of protein right pieces of protein that are secreted when you have an infection you can see there's a big bacterium ne with tadi bacterium එක you can see there are antibodies on the surface or around a small virus you can have antibodies so these antibodies themselves can act as they can cause damage to the bacteria or the virus or they can act as sort of identifying molecules for other bigger white blood cells to come and destroy right then it's like they are sort of marking a target for other white blood cells to come and destroy so imagine antibodies are circulating in your blood they detect a bacteria so they bind to the bacteria so when there's a white blood cell going nearby it is going to be attracted to that bacterial cell and it's going to be destroyed it's like marking the bacteria to be destroyed got it antibodies right that's how they neutralize also on in other sort of functions bacteria they produce toxins bacterial toxins they produce these antibodies can directly neutralize those bacterial toxins as well okay so these antibodies they have a sort of a universal structure මෙහෙම sort of a නිකන් කකුල් තුනක වගේ structure එකක් තියෙන්නේ right so here and here they can bind to these viruses bacterial toxins bacterial sort of the cell wall and they can sort of mark the sort of the foreign substance to be destroyed right antibodies immunoglobulins they are proteins අපි immunity කරනකොට we are going to learn about those in detail හරිද there's another one called receptor proteins right so they are signaling molecules අපි එතන ඒක ඉස්සරහින් ලියා ගන්න okay right here receptor proteins in synapses receptor proteins මට ඉඩක් තියෙනවා right receptor proteins found in receptor proteins in synapses there is something in the nervous system synapses. Okay, synapses. You have heard of things, ne? Dopamine, serotonin, අරම ඒවා අහලා තියෙනවනේ. Those are substances called neurotransmitters that are involved in nerve signal conduction. Okay, ඔය ටික තමයි. Quickly summarize, there are several functions, but all the functions of proteins you can sort of condense into these set of functions. There are enzymatic proteins, structural, storage, transport, hormonal, contractile and defensive. What I want you to do is visually remember the diagram with the function. Okay, enzymatic proteins, they can break bonds or make bonds. Storage proteins found in the egg and the milk. The defensive proteins, they can sort of stick to bacteria or virus. Transport proteins, they can transport substances in blood or into cells. Then structural proteins, keratin, collagen. Yeah, ඒ වගේ. That's why I gave this set of diagrams. හරිද? Okay, අපි let's take a break, no? 15 minutes. Okay, any questions? මොනවද තියෙනවද question there? No? Okay, let's take a small break. Million people, small break. Okay. don't think in any other class they have gone this far. In other classes, I don't think they have gone this far. But anyway, we are working to a program, right? So it doesn't matter where the other classes are, right? Okay, right. So we'll continue, right? So the plan today is I wanted to cover until the structure of DNA, right? So we'll give a try, right? Okay. So shall we move on? Any questions? Online people, let's start. Okay. Right. So another news for you, if you are missing the class, physical people, right? Anytime, I mean, if you have missed the class, missing the class, you can request the recording, right? For the first few months, I'm going to send the recording to the group, right? Every week, I'm sending the recording to the group, right? But as the time goes on, I won't be sending the recording to the group. But if you are getting absent, you can of course request from the hotline. Okay, I was absent on this day, so you will be sent the recording as soon as possible. Okay, and also my recordings are they are unlisted in YouTube, so there won't be any sort of extra data charges. Google Drive දාගෙන එහෙම දඟලන්නේ, right? So it's going to be unlisted. So from anywhere you can just watch them with minimum data charges, right? ඒවත් ඉතින් ප්‍රශ්නනේ, right? So without any lagging issues or quality issues or nothing, right? It's YouTube videos that is shared personally with you, right? YouTube videos that are shared only with you. So you have that luxury. So online people, they of course they can access from the website, right? For you, you can directly request a recording, and anytime if you are getting absent, you can watch, right? So that's a luxury that you have. Right. Anyway, so okay, let's move on to nucleic acids, right? Any questions? No? So in nucleic acids, right? So all these things are proteins working. I mean, they are the workforce. Ne? If you take a cell, the proteins are the workforce. Ne? So you can see there are so many. They can act as structural, transport, regulatory, enzymatic, hormones, right? So many functions are there. So then they are the working people, right? But in any working any workforce should have a controller, should have a master or a sort of someone who is in control, who is giving the instructions, giving the orders. Yeah. So that's what we are going to learn at the end of all these molecules, the final group of molecules that we are going to learn. That is the molecule or the group of molecules that are in control. They are the molecules that are going to store all the information that is needed to sort of, to decide how the cell behaves, what sort of a cell it is, and what are the things done by that cell. Right? Everything is going to be determined by this sort of data. These are data molecules and they are controlling molecules. They are nucleic acids. Out of the nucleic acids, the sort of the if you take on earth, every living cell has DNA as their what we call as a genetic material. Genetic material is the way that you store information that will be transmitted from one generation to the next, right? I have told this previously also, right? If you take any living cell on earth, any living cell, bacterial, algae, fungi, right, animal, plant, whatever cell, they have 4 characters that are common. Harida? All cells are going to have a plasma membrane. Harida? Then all cells are going to have what? Inside you are going to have a protein making factory that are called ribosomes, chutu ribosomes. Okay? Then what else? All cells are going to have DNA as the nucleic material or the genetic material, and finally they are going to have a gel like material inside the cell that is called what? The cytoplasm. Okay? So those things are shared by all living cells. Those things are shared by all living cells. So today I think let's try to cover as much as possible up to the formation of polynucleotides at least, right? I don't want to rush this theory a lot. Okay, so let's see. As always, you know, we are learning about polymers, no? Right? We are going to learn about polymers. So as always, if you are going to learn about a polymer, you should start with the monomer. If you are going to learn something complicated, we should always start with the building block. Yeah. So let's read page number 19. Nucleic acids are polymers existing as polynucleotides made up of monomers called nucleotides. Okay. So nucleic acids are biopolymers. Okay, idea එක තියෙන්න. What is a monomer? Nucleotide. They contain carbon, hydrogen, oxygen, nitrogen and phosphorus. Highlight the molecular composition. Highlight the molecular composition. Nucleic acids are macromolecules and biopolymers. Okay, so that is also important. Nucleic acids are macromolecules and biopolymers. There are 2 types of nucleic acids, DNA and RNA. Okay, DNA and RNA. DNA, what is the full name of DNA? Deoxyribonucleic acid, and RNA is just ribonucleic acid. Okay, right. So we are going to study the structure of a typical nucleotide. They say structure of a nucleotide. Nucleotides have 3 components, namely pentose sugar, nitrogenous base and a phosphate group. Okay, pentose sugar, nitrogenous base and a phosphate group. So if you take a nucleotide, okay, if you take a nucleotide, there's a pentose sugar, right? There's a pentose sugar. Carbon පහයි 1 2 3 4 and 5 okay. Connected to this first carbon, you have a group called a nitrogenous base. අපි යට කියමු N base කියලා. Nitrogenous base. හරිද? Connected to this fifth carbon, මෙහෙම fifth carbon එක තියෙන මේ කොනේ, හරිද? දැන් මේ this is all these corners have a carbon නේ. අපි අඳින්න නැති වුණාට these corners indicate a carbon joint නේ. A carbon junction. දැන් මෙතන oxygen එකක් තියෙනවා. Oxygen 1, 2, 3, 4, 5. At the end of this sort of bar, you have a carbon atom there as well. Well, connected to the fifth carbon, you have a phosphate group, which we sort of indicate like this. This is a nucleotide. This is a nucleotide. Okay. This whole thing is a nucleotide. If you didn't have this phosphate group, මේ phosphate group එක නැත්තම් අපි යට කියන්න පුළුවන්, that means just the pentose sugar and the N base, අපි යට කියනවා nucleoside එකක්. nucleoside එකක් කියලා කියනවා. Okay. So අපි ටක් ගාලා මේක label කරමු. Right. This is the pentose sugar. This is the nitrogenous base. ඔයගොල්ලෝ එක label කරන්න. This is the phosphate group. Okay. Nothing much. Then අපි දන්නවනේ අපි monosaccharides ඉගෙන ගත්තා. We learned about the triacyl glycerol. We talked about the amino acid. Then අපි ඉගෙන ගන්නවා nucleotides ගැනනේ. Monomers. Then දන්නවනේ මෙතන මේ space එකේ it is designed to, the space is given for you to draw this. Okay. The structure of a nucleotide. I have given on the side, left hand side, the proper structure with the molecules. ඒක ඕනේ. You only need to be able to draw this symbolic form. හරිද? Nucleotide. To have this idea clearly, the phosphate group, if it's missing, you end up with a nucleoside. Okay. Then let's talk a little bit about these pentose sugars. Right? Pentose sugars are of two types. Okay. If the pentose sugar is ribose, what do you call the nucleic acid that you end up with? It is ribonucleic acid, no? Right? Ribose. You can also have a form of ribose that is called deoxyribose. Deoxyribose කියලා කෙනෙකුත් ඉන්නවා. Got it? I have given the deoxyribose and ribose. මේ බලන්න යටම in this page. What is the difference between deoxyribose and ribose? You can see in deoxyribose, connected to the second carbon, there is no OH. There is only one H. Check the diagram. There is only a H. One oxygen is missing. So it's a deoxy. One less oxygen ribose. That is the only difference. So there are two types of pentose sugars as deoxyribose and ribose. So that is given. Okay. Then we move on to nitrogenous bases. Nitrogenous bases right? There are two types of nitrogenous bases as purines and pyrimidines. Purines and pyrimidines. Purines and pyrimidines. Okay. So, you need to be able to compare purines versus pyrimidines. Purines are composed of 2 carbon skeletons. They have 2 carbon skeletons, and usually pyrimidines have only one. Okay. Purines are larger with 2 carbon skeletons. Pyrimidines are smaller with only one. Right? We can say 2 carbon rings. This one has only 1 carbon ring. Okay. This one has only 1 carbon ring. So, purines, you have 2 types of purines that you need to know. Purines are the dekak that you need to know. Okay. Adenine and guanine. Adenine and guanine are the purines. Okay. Pyrimidines, you have 3: cytosine. Very careful, this is thymine, 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 and uracil. Uracil. Pyrimidines 3, purines 2. Okay. I have given all. Write down, there's 2 dotted lines. Ne? In purines, there are 2 types, namely adenine and guanine. I have given in the diagram. In pyrimidines, there are 3: cytosine, thymine, uracil. All these are indicated by symbols. We are going to quote symbols there. Adenine is by A. A for adenine, A for apple, bio A for adenine, G for guanine, C for cytosine, T for thymine, U for uracil. Okay. U for uracil. Okay. Clear. So, those are the building blocks. So, you can have a variety, right. So, imagine, so මෙයා විතරනේ change වෙන්නේ නැත්තේ phosphate group එක විතරයි. So, you can have ribonucleotides how many times? You can have how many ribonucleotides? Okay. This uracil and thymine are 2 special people. Okay. Thymine you can find only in DNA. Only DNA. Thymine only found in DNA. Uracil only found in RNA. Thymine only in DNA, uracil only in RNA. So, with that in mind, okay, so with ribose, what as nucleic acid do you make? Ribonucleic acid. What are the types of ribonucleic acids you can have? You can have ribonucleic acids with adenine, guanine and cytosine and uracil. Uracil. How many, what are the deoxyribonucleic acids you can have? Sorry, ribonucleotides, I am sorry. Deoxyribonucleotides you can have with adenine, guanine, cytosine and thymine. For RNA you have 4 types of nucleotides. For DNA you have 4 types of nucleotides. The only difference is what? In DNA you have thymine, in RNA you have uracil. Them jolly again, right? Isarata you will completely remember this without a problem. Okay? So now you know the basic building block. That is called the nucleotide. Clear? Now we can move on to the polymerization. Ekama story and amino acid igena gatta, polymer igena gatta, right? Ekama story again. Got it? So what's going to happen is, I'll erase this side. I'm introducing you a new bond, such as the peptide bond, glycosidic bond, the ester bond. There's a new bond that is called the phosphodiester bond. Phosphodiester bond. Kila alut bond. I'm going to introduce. Okay, right. So let's read formation of nucleic acids. Page 19, formation of nucleic acids. Millions of nucleotides joined by phosphodiester bond to form polynucleotide chains. By condensation between the OH group of the phosphate of one nucleotide with the OH attached to the third group of the pentose sugar of the other. This results in a backbone with repeating pattern of sugar phosphate units. Okay, right. Now, make a but you need to be able to explain between what and what you have the bonding. So, you have a pentose sugar like this. This is your pentose sugar 1, 2, 3, 4, 5. Connect to the 5, you have a phosphate group. Here you have an OH. There's an OH there. Okay. Then I'll draw this. In the next phosphate, I mean in the diagram I have given it O minus. O minus when originally it is supposed to be OH. Okay, it was a method connected to the carbon, and here also you have a O minus. Okay, that doesn't matter, right? So similarly, metanath ehema thamai story, right? I'll draw it in black color, right? So pattern. Okay. So this is the phosphate group of the next nucleotide. Anith nucleotide dege, this is the phosphate group. One, 2, 3, 4 and 5 ne. Method inno N base. Okay, there you have the N base. Okay, so now what we are going to do is we are going to connect these two. So when that happens, you are going to have a completely new bond, right? That is called what? This OH and this hydrogen goes away, okay? And there is going to be a new bond that is forming between this phosphate and the third carbon, that is called what? Phosphodiester bond. Phosphodiester bond. Okay, it's a new bond that you are learning. Phosphodiester bond. ඔය විදියට මෙහෙම දිගටම යනවා between the third carbon and the phosphate group, right? මම මේ විදියට symbolically අඳින්නම් phosphate, right? Third carbon of the pentose sugar and the phosphate group, you are going to have this sort of formation of a condensation reaction happening. This is resulting in a chain, no? It's a polynucleotide chain. Several nucleotides that are connected by phosphodiester bond. Okay. Then what do you notice here? Then this is fifth carbon. This is fifth carbon. 1, 2, 3, 4, 5. Yeah, that is the fifth carbon. Then what is the carbon that is sort of pointing outwards? The third carbon. No? The third carbon ready to form a new bond. Yeah, the third carbon ready to form a new bond. This bond formation, okay, this bond formation always happens from the 5 carbon with the at the top and the third carbon at the bottom. So we call this bond formation happening in a direction that we call as 5 prime end. මෙයාට කියනවා 5 prime end එක කියලා. මෙයාට අපි කියනවා 3 prime end. Okay, always we say therefore phosphodiester bond formation එහෙම නැත්නම් polynucleotide formation happens from 5 prime end to 3 prime end. තේරුණා? 5 prime end to 3 prime end. That is how polynucleotide chains occur, form by formation of phosphodiester bonds. Again, if I read, right, between the OH of the phosphate and with the OH attached to the third carbon, may phosphate guy, this third carbon, you are going to have a phosphodiester bond. Clear? Okay. Then they say this results in a backbone with repeating pattern of phosphate units, sugar phosphate units. මොකද මේ backbone එක කියන්නේ. දැන් මෙතන ආපහු backbone කතන්දරයක් අල්ලගෙන. අපි මේ night I'll draw this nitrogenous base in a different color, then it is easier, right? So again we are talking about a backbone. You can see this is a phosphate, sugar, phosphate, sugar, phosphate, sugar, right? So they are not on the side, but we say they are like one after the other. Sugar, phosphate, backbone. So this whole section, when we set what? The sugar, phosphate, backbone. Sugar, phosphate, backbone. Okay, right? So this is only one chain. Then I erase the nitrogenous bases because I am going to draw the nitrogenous bases in a different way, right? Because these nitrogenous bases I am going to draw like this in their proper form, right? As the rings, they are ring form. Okay. Okay. That is what? Just a pyrimidine. These are two purines. This is just these are purines. This is just a pyrimidine. Okay. So what is the backbone? The sugar and the phosphate group. They are connected by phosphodiester bonds. That forms the backbone of this structure. So you have only one. 5 prime to 3 prime, and there's one chain going downwards. Where is the other one? Why do you need another one? Okay. Nucleic acids are linear polymers of nucleotides. So you can see they are linear molecules. There are two kinds of nucleic acids depending on the type of sugar molecules involved. If the sugar molecule in the nucleotide is deoxyribose, then the nucleic acid is DNA. If the pentose sugar is ribose, then the nucleic acid is RNA. These are simple things, no? Okay. DNA contains adenine, thymine, guanine, cytosine, and RNA contains adenine, guanine, cytosine, and uracil. Can you highlight the difference? DNA has thymine and RNA has uracil. What differences they can please highlight? Highlight those two differences. So what is this? It's a phosphodiester bond. Hi, right? Then this is just one polynucleotide chain. When we are going to talk about DNA, we need to have two polynucleotide chains. එකක් විතරක් මදි. DNA ට තනියෙන් ඉඳලා පුරුදුම නැහැ. Okay? So we'll quickly move on to the structure of the DNA molecule. Okay? Structure of the DNA molecule. Right? Then we'll come back and discuss this space here. Okay? Alright. Structure of the DNA molecule or the Watson and Crick model. Okay? DNA molecules have two antiparallel polynucleotide chains that spiral around an imaginary axis, forming a double helix. Okay. Right. So DNA is, I think, one of the most famous molecules in the world. Ne? Anyone, I mean, molecule idea එකක් කිසිම නැති කෙනෙක්ට උනත් DNA එකක් වගේ ළඟින් එකක් කියවන්නුවම කියයි මේ DNA කියලා. From the චූටි කාලේ ඉඳලා we are exposed to this molecule shape, right? You know, sci-fi movies and everything, you have DNA, you know, right? So actually this structure was, I mean, the experiment to identify this structure was not done by Watson and Crick. There was a female scientist called Rosalind Franklin. There's a technique called X-ray crystallography that we use to this day to identify the structure of molecules. Okay? Using X-rays, you can identify molecular structure. I mean, you can visualize. So for the DNA molecule, that technique was done by Rosalind Franklin. Okay? And she had some findings. Watson and Crick went through her findings and they put forward the idea, okay, this is the structure of the DNA molecule. They gave it a name, it's a double helix, and they described it. They described it and they presented it. Okay? So the credit of sort of presenting the idea of the double helix goes to Watson and Crick. But the actual technique was done by a different scientist. Okay? Anyway. So talking about DNA, they say it has two antiparallel polynucleotide chains that spiral around an imaginary axis. අපි ඔය spiral කතාව පොඩ්ඩක් තියමුකෝ. Okay? It has to have two polynucleotide chains. දෙකක් ඕනෙලු. So how do you sort of put a second one into this mix? You should have another polynucleotide chain running like this. Ne? There should be another polynucleotide chain running. But what is the key word? It's antiparallel. Antiparallel කියන්නේ මොකක්ද? Right? Antiparallel කියලා කියන්නේ මෙයා යන්නේ from 5 prime to 3 prime up ඉඳලා down නම් the other chain should go from bottom to top. Okay? So ඒක අඳිනකොට a bit tricky, right? ඒක අඳින්න වෙන අනිත් පැත්තට. So I'm supposed to start from here, right? So there's a pentose sugar like this, and it has a phosphate group. Okay? And it has a phosphate group, and connected to it by a phosphodiester bond, there's another phosphate, and connected to that you have another what? Again, pentose sugar. Okay? You have another pentose sugar, and by another phosphodiester bond you have another phosphate group connected, and then again you have another pentose sugar. Oh my god, David. Got it? So this chain is running in the opposite direction. Right? This chain is growing from bottom to top. Ne? This chain is growing from bottom to top. Sleepy? No, right? Right? This chain is growing from bottom to top. So what is the direction of that? This is the fifth carbon. If I put numbers, this is 1, 2, 3, 4, 5. No? Sorry, 1, 2, this is 3, 4 here, 5. No? That is the 5 prime end. Here, this is 1, 2, 3, 4, 5. This is the third end. No? So this is the 3 prime end. So what is the growth of direction growth? 5 prime to 3 prime. Mayman. Yeah? The growth happens opposite directions. Right? So that is what we call as anti-parallel. Then connect. How do we connect these two? How do we connect these two? Okay? I will draw only one connection. So connected to this pentose sugar, what do you have? A nitrogenous base. Okay? Connected to this pentose sugar, you have a nitrogenous base. The nitrogenous bases are going to join with each other. The nitrogenous bases are going to join with the nitrogenous base of the other chain by hydrogen bonds. Hydrogen bonds. Okay? Connected to this pentose sugar also, you are going to have some sort of nitrogenous base, right? You are going to have a nitrogenous base. Those two nitrogenous bases are also going to be connected by hydrogens. Okay. So basically what you have? You have two polynucleotide chains running in opposite directions. Okay. Therefore you call them as antiparallel, and they are interconnected by hydrogen bonds. They are interconnected by hydrogen bonds. Clear? Got it? Okay. I'll show this with the projector so you have a clear, nice 3D idea. Okay. Let's read. The two sugar phosphate backbones run in opposite directions from each other, and this arrangement is referred as antiparallel. Highlight, this arrangement is referred as antiparallel. This arrangement is referred as antiparallel. Highlight the word antiparallel. Okay. The sugar phosphate backbones are on the outside of the helix, and nitrogenous bases are paired in the interior of the helix. So you can see these are considered as to be outside, and these sugar phosphates are considered to be in the inside of the helix. Then okay, imagine have you seen these spiral staircases? Spiral staircases. On the two sides you have the sort of handrails. No? The two handrails, or the sort of where the stairs are connected, padi connected those are the sugar phosphate backbones. The steps in the middle, those are the nitrogenous bases. Got it? Those are the nitrogenous bases. The two strands are held together by hydrogen bonds between the paired nitrogenous bases. I told you there is so there's hydrogen bonding between the nitrogenous bases. Okay, right. Let's see. I'll use the software, and you can have an idea, sort of a 3D idea, right? So you should be able to draw a nucleotide. That's all, right? Nucleotide, and you should be able to draw a DNA. Not this complicated, but the diagram I have given in the tute, right? The diagram I have given, you should be able to label the sugar phosphate backbone and the nitrogenous base. Simple labeling you should be able to draw the double helix. Okay, that's all you need to do. I don't need to erase, no? There's a screen, right? Right. There's a remote somewhere, right? Well, the technology is provided, right? So, yeah. Let's see if it works. Nice. All right. Online people, can you see? Is there any way to switch off these two lights? Comedy off color. Ah, nice. Okay. Better. Okay. Right. Don't. The wallpaper is interesting. This is a new, not new. This is a patient who had a accident. Okay, road traffic accident. Okay, right. Uh, it's a new technique of CT scanning. Online people, can you see? Right, it's a technique of CT scan that you can create a sort of a 3D view. Okay, you can create a 3D view of the using several CT, several hundreds of X-rays. You can create this 3D view. The frontal bone is fractured, and the upper teeth are fractured. The nose is fractured. This sort of forehead is fractured. Yeah, motorcyclist. Okay, motorcyclist. Lorry right? So he was transferred with this CT. So it has to be repaired by a maxillofacial surgeon. Okay, requires surgery, but the immediate problem, what is the immediate problem he is going to have? Right, risk to life. What is the immediate risk to life? Yeah, the airways are going to be blocked. The breathing can be affected because the nose has going to be severe bleeding, right? Sort of blood can be aspirated, blood can go into the lungs, and so that is the immediate problem. Cosmetic problem, right? Right. Anyway, so we were talking about nucleotides. No? Right. So let's start with the basics. You can see, right? Online people, it's clear enough, neither. Yeah. Hurry. Then Mulimma, let's focus on to these sides. Can you see? Those spheres, those are what? Those are the pentose sugars. Those are the pentose sugars. Right? These are the pentose sugars. I think. Clear? And you can see these are the, I'm sorry, those are the phosphate groups, right? Those are the phosphate groups. These are the pentose sugars. Right? You can see pentagonal shape. Right? These are the pentose sugars. So you can see they are arranged one after the other. So that's why we call it a sugar phosphate, sugar phosphate, sugar phosphate backbone. Sugar phosphate backbone. On the other side also you have the similar thing, right? The thing is these two sugar phosphate backbones run in opposite directions. Okay, whatever 5 prime to 3 prime direction. Then let's go to the center, which is where the nitrogenous bases are. Then if you have only one ring, what is it? Is it a purine or a pyrimidine? One, online people, it's a pyrimidine. It's a pyrimidine. If you have two, purine. දැන් මේක බලලා ඔයගලන්ට කියන්න බෑ. So what are the options for pyrimidine? This is DNA. හරි? This is DNA. What are the options for pyrimidines? Thymine, cytosine. ඔය දෙකෙන් එකක් වෙන්න. දැන් මේක ඔබලා බලමු. There is cytosine. Okay? Then there are two rings. Those are purines. What are the options for purines? දෙකමයි adenine and guanine. අපි මේක දැන් බලමු. This is guanine. So with that I am going to introduce you to a law. දැන් මේක නීතියක්. In biology you can't break this law. Always a pyrimidine binds with a purine. Okay? Always a purine binds with a pyrimidine or a pyrimidine binds with a purine. Pyrimidine set වෙන්නෙත් නැහැ. Purine set වෙන්නෙත් නැහැ. තේරුණාද? පේනවනේ? There's always single ring එක්කෙනෙක් එක double ring. Single එක්කෙනෙක් එක double. Single එක්කෙනෙක් එක double. Never single or double. Got it? පේනවනේ? If you go up and up, it's always single with a double. Always a purine binds with a pyrimidine. Law. Right? Then there's another law. ඒ කියන්නේ නීතියට කැලි දෙකක් තියෙනවා. මෙතන you can see what are these? This is the hydrogen bond. How many hydrogen bonds can you count? 1, 2, 3. නේ අපි දැන් මෙහෙම now it is more clear. No? Sorry. Molecule. Right. It's okay. You can see clear here, no? 1, 2, 3 bonds here, right? So, what is the first part of the rule? A pyrimidine always binds with a purine. The second part of the law is, this is who? Cytosine. Okay? Cytosine always connects with guanine. Guanine and cytosine by 3 hydrogen bonds. Guanine හැම වෙලාවෙම යන්නේ cytosine එක්ක by 3 hydrogen bonds. Okay? Then කවුද අනිත් දෙන්න missing? This has to be red එක මෙයා කවුද එතකොට? Adenine. Adenine always goes with, who is the remaining one? Thymine with how many hydrogen bonds? 1, 2. Okay? Adenine thymine by 2 hydrogen bonds. Okay? Adenine thymine with 2 hydrogen bonds. Guanine and cytosine with 3 hydrogen bonds. Got it? That is what we call as the base pair rule. There's a sugar phosphate backbone on the two sides and nitrogenous bases on the center. They are connected by hydrogen bonds. Okay? So now we are not going to draw the backbone, right? It's the same. No? හැම වෙලාවෙම මෙහෙමයි. ප්‍රෝටීන් එකක් ගත්තත් backbone එක අඳින්නේ නැහැ අපි ඉස්සරහට. ඕම යනකොට we are going to get the spiral shape. Spiral shape එක එනකොට the nitrogenous bases also are going to be like this. So if we zoom out, this is the antiparallel double helix. Okay? So if we go to a different view, this is the classical model of the DNA double helix. Ne? So on the two sides, what do you have? Sugar phosphate backbones, and in the center, what do you have? Nitrogenous bases. Clear, right? Right? Then the DNA molecule. So that is a DNA molecule. That DNA molecule is going to condense and it's going to wrap around proteins. On proteins, whatever wrap, it is going to become even more condensed. Condensula. At the end, you are going to get a structure called a chromosome. Right? You are going to get a structure called a chromosome, which is how DNA is packed into your nucleus. Right? That if you take one of your cells, you have so many DNA. Right? The length of the DNA molecules that you have, you can never pack them in a normal way inside your cells. You need to have very tight packaging, otherwise you are never going to fit all that DNA inside a cell, inside the cell's nucleus. Yeah? So that's why we have this much of a packaging. Then ඒ ටික ඕනේ නැහැ ඔයගලන්ට. මේ ටික තමයි ඕනේ. Got it? The DNA double helix introduced by who? Watson and Crick. Okay? Clear near the base pair rule like, right? What is the base pair rule again? Purine always binds with a pyrimidine. Okay? Adenine and thymine by adenine and thymine දැන් මේ වගේ මෙතන වගේ adenine and thymine by 2 hydrogens bonds, guanine and cytosine by 3 hydrogen bonds. Okay, clear? Any questions? Okay. Okay, right. Clear. Okay, so that makes sense, ned? So base pair rule kiyala there's a small topic. Can you see? Base pair rule. I have given the base pair rule. Always a purine base pairs with a specific pyrimidine base. Always a purine binds with a pyrimidine. That is the base pair rule. Then write down the next part. Yeah. What is the next part of this rule? Adenine binds with thymine by 2 hydrogen bonds. Guanine binds with cytosine with 3 hydrogen bonds. Dotted lines දෙකක් තියෙනවා නේද? Yeah. Okay. Hence, the two strands of the chains are said to be complementary to each other. These pairs are known as complementary base pairs. In this original double helical structure, each complete turn consists of 10 base pairs as shown in the diagram. Okay, can you see? One complete turn means, okay, that means 2 twists, right? That means the sort of the staircase has turned 360. Okay, the staircase has turned 360. That means one complete turn going to contain what? 10 base pairs. Okay, highlight, consists of 10 base pairs as shown in the diagram. In one full turn there is going to be 10 base pairs. One full turn, 10 base pairs. Then functions of DNA you need to know. Store and transmit genetic information from one generation to the next generation. Store the genetic information for protein synthesis. This protein synthesis part we are going to study next week. Okay, how DNA, how the information present in DNA is used to make a protein. Okay, then our cake example එකම තමයි. Imagine the DNA is like the recipe book. Okay, how to make a cake කියලා there's a recipe. By looking at the DNA recipe, you are going to make a protein, which is a cake. Okay, that we are going to study not next week, the week after, when we are studying RNA. Okay, RNA are the main sort of servants of DNA that are involved in making proteins. Got it? Any questions about DNA? In this space, okay, in this space, I want you to draw the DNA double helix method. Okay. That is the one that is given here. There is one in your resource books as well. Okay, you can draw the resource book diagram or the diagram I have given. Anything is fine. Okay, clear? So, look into getting the resource book as soon as possible. Okay, I mean I can print and give it to you and as part of my tude, but why? I mean, it's useless because I feel better that you have your own personal copy of the resource book. Okay, you should have your own personal copy of the resource book that you highlight, that you write on, that you draw on, whatever. It's your own personal resource book. Okay. Okay. Draw the DNA double helix. So it's basically like this. There's the okay, my drawings are not exactly the best. Okay. So you have to clearly draw the sugar phosphate backbone. Okay, clearly draw the sugar phosphate backbone, and in between you have to draw the nitrogenous bases. So black අපි ගමු guanine කියලා. Blue let's take as adenine. You can draw, you can use whatever color you want. Okay. Okay. Adenine always binds with thymine. Adenine always binds with thymine. Guanine binds with cytosine. Guanine binds with cytosine. And indicate the number of bonds. Okay. Adenine binds with thymine by 2 hydrogen bonds. Guanine and cytosine by 3 hydrogen bonds. Adenine, thymine 2, guanine, cytosine 3. Okay. ඒ වගේ draw a full double helix. Label the sugar phosphate backbone. මේ දෙන්න මොනවාද? These are sugar phosphate backbone. Sugar phosphate backbone. That's it. Okay. Draw one spiral for now. Itu ටික අපි ගෙදර ගිහලා අඳින්න. Okay. So, didn't I give a essay on carbohydrates? Functions of polysaccharides, didn't I give? Briefly describe, as a short note, briefly describe structure and functions of polysaccharides. Do not. Okay. Then take down homework. Essay writing book එක තියෙනවා නේද? Right? This is the second one we are going to write. Okay. අපි ලියමු short note එකක් as homework. New people. Okay. The only book you need to bring to the class is a CR book to write essays. Okay. Online people also new one, new people. The only other book you need to bring, other than the resource book, the tutors and the essay writing book. Okay. So we have written already one. Water essay got liver. Right. So this is the second one. This is a short note. I am introducing you to short notes. Right. Structure and functions of polysaccharides. 20 points. Okay, 20-point short note there. So you might be thinking, why only 20? Because in a short note you have part A, B, C, okay, right? So all together, all three parts, you are going to have about 50 marks. Okay, then someone make a 20 in the others can be 15 දෙකක් වෙන්න පුළුවන්. So ඒ දෙක අදාළ නෑ. I'm only going to give you one part, only a part of the short note, that is structure and functions of polysaccharides. Okay, මොනවාද නීති දැන් මේකේ? Short note game එකේ නීති මොනවාද? You can write in point form. You don't need to write full sentences. Okay, you can write in point form. හැබැයි නිකන් තරු දාලා මල් දාලා ඒව කරන්න එපා. Just dot දාගෙන ලියාගෙන යන්න. Okay, dot ඕනෙත් නැහැ. One rule after the other, write down. Okay, no need sentences, just points. Okay, ලියලා බලමු how it's going to turn out. Okay, homework, structure and functions of polysaccharides. Online people, right? Clear? Short note homework, structure and functions of polysaccharides. Okay. So next week it's going to be the practical session, and the week after we are going to finish organic molecules. RNA ටික ඉවර කරලා organic molecules ටික ඉවරයක් කරනවා. Then we are going to start the brand new lesson, not the lesson, the new segment, that is cellular structures. Okay, so we are going to cover the first part, that is microscopes, in the practical session, and then we are going to move on to cell theory, structure of the plasma membrane, aroma. Okay, hold on 5 minutes here, right? In the cool down paper 2, there were some questions we didn't do, right? Cool down 2 ekේ there were some questions we couldn't do, right? Mogo tired? Yeah. एक MCQ एक कर लें. Okay. Cool down paper 2. Question number 5. What is the, select the correct statement about proteins. Okay. Cool down to MCQ 5. Tag gala kan ba? Okay. I will send the cool down paper 2. All are available in the groups. Okay, I sent to the chat. Okay, quickly download and do question number 5. Finish, ne? You didn't get to? So it's a one-man show today, right? So what's the answer? Second. Okay. Anyone else? Select the correct statement about proteins. Over again. Accumulation of weak acids and weak bases in the body causes for protein denaturation. Is it going to cause denaturation? Any weak acid no. Right? In a weak acid, the proteins are good. Ne? Amino acids are going to act as buffers. Okay. So you can say in the presence of weak acids and weak bases, amino acids act as buffers. Amino acids act as buffers in weak acids and weak bases. Okay, amino acids act as buffers. Too cold? Too cold? Then it right? Okay, statement 2. Secondary structure of protein is caused due to the formation of intermolecular H bonds in the alkyl group in the phospholipid chain. Blah blah, right? Again, it doesn't make sense at all, right? So secondary structure of protein, correct what is the correct way? Caused by the formation of intramolecular hydrogen bonds. Right? Intramolecular hydrogen bonds, alkyl group, and right? There's no such thing as a phospholipid chain, right? Yeah. Maximum number of gas molecules that can be transported in hemoglobin is 8. Gas molecules. Can you take 8 molecules? No, ne? 4, ne? 4 molecules you can transport. Alpha helical structure of secondary structure can be found in chitin. What is chitin? It's a polysaccharide. No? Chitin found in fungal cell walls and arthropod exoskeleton. It's a protein. Right? No, right? Chitin is a polysaccharide. Maybe a spider silk confuse chitin arthropod right? Put a lava confuse 5. Amino acids are considered as neutral as it contains acidic properties as well as basic properties. Yes. Amphoteric, no? You can write down in front. Amphoteric. Mention an example for following compounds page number 2. DNA a protein with intra and intermolecular bonds a protein that neutralizes foreign bodies. A protein with intra and intermolecular bonds and a protein that neutralizes foreign bodies. Yeah. Then when they say a protein with intra okay. Secondary structure level you have intra. When they say inter, what should come to your mind? It's a quaternary structure. You have multiple polypeptide chains. 2 or more. That's why you have intermolecular. Yeah. So a protein with inter and intramolecular you can write maybe collagen, hemoglobin. Yeah. Collagen, hemoglobin. Then a protein that neutralizes foreign bodies. Yeah. Immunoglobulins or antibodies. Okay. Immunoglobulins or antibodies. Okay, that's it. So, see you all next week for the practical session, and ask your friends to come. It's a free session. Okay. And so, you will be given the new tute together with the practical guideline. And any questions? No. Go. The next class is about to start. Online people, any questions? All clear? All clear?